Welcome everyone to the Reach Out Science Slam Finals. Before we get started, if you would like to access captions for this event, you can find a link to live captions in the YouTube description, and there's a QR code here on the screen you can use to access the captions link. All right, the big day is finally here. It's the Reach Out Science Slam Finals. I'm Megan from the Museum of Science. My pronouns are she and her, and I am so excited to be your host tonight. Now, before we get started, I'm honored to be joined by the president of the Museum of Science, Tim Ritchie, who has a very special welcome for us tonight. Over to you, Tim.
Thank you, Megan, and welcome everybody. It's a real honor to be here with you for the Reach Out Science Slam Finals. You know, everyone loves a good story, and the stories of science are stories of awe and wonder. There's stories of persistence and hope. There are stories of shocking truth. And those are the kinds of things and the stories that we get to tell here at the Museum of Science. Our mission is to create in everyone a lifelong love of science. And to what end? So that we can build a world where science belongs to each of us for the good of all of us. Each of us, all of us, everyone loves a good science story because it's a story about truth that needs to be told. And what better time than now? What more important time than now? Where have you ever seen a time when public trust in science was more important? So good luck to you tonight, all of you who are in the finals. Have a good time, you be you, and thank you to the judges for giving your time for tonight's important event. Megan, back to you.
Great, thank you so much, Tim. That was wonderful. Now, our event tonight is part of the Reach Out Science Communication Challenge, a Museum of Science program designed to connect scientists with public audiences. And it's been quite a journey for the scientists who will compete tonight. They started out this past winter taking part in our Reach Out Science Communication workshops. Then they entered into the Reach Out Science Slams. They had a chance to attend coaching sessions with our Museum of Science team. And all of this led to the amazing semifinals performances that earned them a spot here in the Reach Out Finals. We've had a blast working with all of them, and we cannot wait to see them all present for us tonight. Now, this evening, 2 of our 6 Reach Out finalists will win $1,000 prizes, and one of these winners will be selected by you, our public audience. So stick around and we'll tell you how to vote for your favorite science slammer towards the end of the program. Now, the other winner tonight will be chosen by our fantastic panel of expert judges. Joining our judging panel for the Reach Out Science Slam Finals, all 4 of our outstanding judges are here tonight to support our 6 finalists and ultimately make their choice for the grand prize winner. Raven Baxter, also known as Raven the Science Maven, is a molecular biologist, expert science communicator, and has been featured in Fortune magazine's 40 Under 40 list for 2020. Melissa Cristina Marquez is a shark scientist, TV presenter for National Geographic and the Discovery Channel, and author of the upcoming book series Wild Survival. Emily Calandrelli is an aerospace engineer turned science communicator, Emmy nominee, and host of the Netflix show Emily's Wonder Lab. And to round out our judging panel, Jorge Cham is the creator of the webcomic Piled Higher and Deeper, or PhD Comics, and he's co-creator of the PBS show Eleanor Wonders Why. Welcome to all of our judges. All right, welcome Raven, Melissa, Emily, and Jorge. We are so excited to have all 4 of you here. Thank you so much for joining us. So, are you ready to hear from our finalists?
Oh, yeah.
Yes.
Yeah, I'm excited. It's been fun to watch the journey leading up to tonight, so I'm sure the pressure is on, but from what we've seen so far, they are all so extremely talented, and I can't wait to hear some science stories.
Great. Thank you. All right. Well, I'm sure that everyone at home is very excited too. So let's go ahead and meet our slammers. Our Reach Out Science Slammers came from all over the country and are each part of one of the 12 National Science Foundation Science and Technology Centers. These are interdisciplinary research centers, which means a bunch of different kinds of scientists come together to discover something new about the world or solve some big challenges. They're working on all sorts of areas like microbiology, artificial intelligence, evolution, quantum physics, cell biology, computer science, and more. These 6 Slammers were selected by our judges and by our audience to compete in the finals today for our top prizes. Okay, our Reach Out finalists come from 5 different centers. First, let's say hello to Sabrina from the Center for Dark Energy Biosphere Investigations. Now next, we have Giska from the Center for Engineering Mechanobiology. Welcome, Giska. And from the Center for the Study of Evolution in Action, please welcome Jolene. And next, let's meet Megan from the Center for Cellular Construction. Hi, Megan. And finally, let's welcome Christina and Ben, who are both from the Center for Integrated Quantum Materials. All right, welcome to all of our finalists. We are all very excited to hear from you. Now, if you would like to cheer on our Slammers from home, you can send a comment at any time by going to pollev.com/Museum of Science, or you can text Museum of Science to 22333 and then send your comment. And we'll keep that information handy on the screen for you. And we'll share some of those comments a little bit later. So go ahead and send some encouraging notes to our Slammers, but please don't cast your votes just yet. We'll let you know when the audience voting opens a little later in the show. All right, now let's get started with our first Slammer of the evening. Our first Science Slam finalist tonight is Jolene DeLima, who is a recent PhD graduate from Michigan State University and is affiliated with the Center for the Study of Evolution in Action, which brings together biologists, computer scientists, and engineers to study evolution as it happens and apply this knowledge to solve real-world problems. Welcome, Jolene.
Hi, I'm Jolene. Welcome to my garden back home in the tropics. These are all pictures of flowers from my garden. Growing up and having all this diversity just outside my front door was amazing. But of course, as you know, diversity is not exclusive to my garden. We see it everywhere. There are just so many different kinds of living things. What is fascinating, though, is that all this biological diversity has one common origin story: evolution. Evolution is the process by which living things gradually change over time. And this story is the same for all organisms. We all descended from one common ancestor. The simplicity in this complexity is delightful, and was especially so for a lazy student who was elated that she did not have to memorize a new story for every new organism. I mean, can you imagine the horror? My mind, though, was blown when I realized that not everybody thought this way. Students describe evolution differently in plants or animals, in something familiar like a cheetah, or something unfamiliar like a salamander, or in humans compared to all other organisms. And they do not see the bigger, simpler picture of how it's all just the same story, and end up having the wrong idea that each organism has its own origin story, and thereby creates so much more mental work for themselves trying to remember it all. And this is a big problem. You cannot truly understand biology or pandemics or life itself without understanding evolution. So now I try to figure out what college students think about evolution. I ask them how they think cheetahs evolved to run faster or humans to stand upright. They often tell me It's because cheetahs needed to run faster, or humans wanted to stand upright. But that's not correct. Faster cheetahs and upright humans probably had better access to food, which meant they lived longer and therefore had more babies than slower cheetahs and non upright humans. Evolution works the same way in all organisms. Understanding the patterns in students' thinking and mistakes will help us greatly improve how we teach evolution. And hopefully, when students see all this diversity around them, while they're perhaps walking through a garden like mine, they will not only appreciate the complexity in the diversity, but marvel at the simplicity that unites it all. Thank you.
All right, let's hear it for Jolene from the Center for the Study of Evolution in Action. Thank you, Jolene. Wonderful job. Now, let's hear from a couple of our judges. So, Jorge, do you have any feedback for Jolene?
Yeah, that was wonderfully diverse, and it evolved into a really good presentation. So a fantastic job. No, I think really it was great. You did a fantastic job. I felt really connected to you. I really liked the way you kind of started it as a story, as a personal story, and I loved the way you connected with the audience and how you were very deliberate in your pacing and your gestures. And I love that time that you like went up close to the to the camera. That was a really nice touch. So great job.
Great, thank you. Raven, how about you? What did you think?
I loved your enthusiasm and how you emphasized the different words in your story. It kept my attention the entire time. And I felt like I felt like I was with you on a journey and we were going somewhere very fun. That you did a wonderful job. Well done.
Great,
Thank you.
Thank you, Jolene. Thank you, judges. Well done, Jolene. Now let's take a look at slam number 2. Next up, we have Christina McGann, a postdoctoral fellow at Mount Holyoke College and part of the Center for Integrated Quantum Materials, which is working to build new atomic scale materials with extraordinary properties. Take it away, Christina.
Imagine that you are driving your car or pedaling your bike down the road. The sun is shining and it's a warm day. But up ahead, you see it. A big pothole. You have to slow down so you don't damage your car or hurt yourself on your bike. In my research, I fix potholes, just not the ones in the road. I fix electrical potholes. This electrical equivalent of potholes in materials makes electricity move slowly through the materials at certain spots, which makes electrical devices made of these materials, like phones and computers, not work well. We can see potholes in the road, but we can't see these electrical potholes with our eyes or with a light microscope because they are 10 times smaller than the thickness of a strand of your hair, or even smaller than that. So we need something that can sense smaller objects. But how can we figure out information about an object, like this mask, if we can't see it with our eyes? We can touch it with our finger to figure out how rough it is or how it's shaped. What I use in my work, and that's me, is an atomic force microscope, which has a tiny finger, 1,000 times smaller than the thickness of a strand of your hair, or 100 times smaller than the potholes. This tiny finger can measure things like roughness and shape, but also things that our finger can't measure, like how electricity is moving through a material. So I use this tiny finger to find electrical potholes that make electricity move slowly in plastics and in two-dimensional materials. These are like sheets that are a single atom thick, so super thin compared to this paper. Once I find these electrical potholes, I can figure out why they're forming, and from there, how to get rid of them. In my work, I bake the plastic by heating it up in an oven in order to cover the material with a layer that fills in the electrical potholes and also doesn't let new ones form. Now, the material is free of electrical potholes and electricity can move faster through the material. With my improved materials, scientists can make better devices, like phones and computers, and even better solar cells that take more light from the sun and turn it into electricity. Thank you.
All right, let's hear it for Christina from the Center for Integrated Quantum Materials. Thank you, Christina. Nice job. And our judges are back too, so let's see what they thought. So Emily, let's start with you this time. Any thoughts you'd like to share?
I love this so much. Christina, I loved your props. I had noticed that you added some new ones and some upgraded props, and I love that. And you also added, I think, some new examples of real world consequences of solving this problem, which I loved how that connects to, why it's important that you're doing what you do. And I think so much of science communication is just grabbing someone's attention and being able to show them something different, like a story told through props, like you did, was so attention grabbing. You did a really good job.
Thank you.
Thanks, Emily. Melissa, anything to add to that?
Yeah, I really loved how you moved about the camera space. You used different props. You really captured us in. So amazing job.
Thank you so much.
Great. Thank you so much, judges. Thank you, Christina. Wonderful job. Now it's time to move on to slam number 3. Our next slammer is Giska Ramadita, a student at Washington University in St. Louis, representing the Center for Engineering Mechanobiology, which aims to understand and harness the power of mechanics in tissue, cell, and molecular behavior across the plant and animal kingdoms. Over to you, Giska.
Do you know what organ this is? Yeah, this is a heart, the beautiful organ that keeps beating every single second of our life. When you are walking, eating, sleeping, or playing. The beating of our heart is a result of crosstalk between the electrical signal and mechanical pumping function. What electrical signal? Yeah, as illustrated as yellow dots here, that's electrical signal inside your heart. So now you can see it's going up when it was running from the upper part to the lower parts. Simultaneously, the heart starts to contract and pump out the blood. Have you ever wondered what if their communication doesn't work well? It will get out of sync, right? The tissue loses its rhythm. This disease is called arrhythmia. Do you know anyone who suffers from arrhythmia? Maybe you do, because it represents a major worldwide public health problem. One case in every 5 deaths in the world. In 10 years, it's estimated 12 million people in the US will have this disease. And the common cause of arrhythmia is electrical signal problem. But that's not the whole story. There can be other causes too, which one of them is related to the structure of the heart. Let's talk more about it. The right part's function is to pump the blood to the lungs, the closest organ to the heart itself. while the left part is responsible to pump the blood throughout our whole body. Sounds like a much bigger job, right? This is why the left part has much thicker wall than the right part, to be able to make bigger pressure to send all of the blood supply to all of our blood vessels. And now, what if the heart wall is too thick? Let's see from this rubber ball. It's so flexible, easy for me to squeeze it. How about this one, a thicker one? It's hard for me to squeeze it. It's too stiff. And this could be what happened to the heart with certain disease, which is hypertrophic cardiomyopathy. When the blood pressure is high, the wall is getting thickened. So you can see it may fail to pump enough blood to our body. It can be a severe problem for young and apparently healthy athletes too. In our group at WashU, we are interested to learn how the stiffness could also be a trigger for heart disease. By looking closely at the biological mechanism, we hope we have a better idea and we can propose a better treatment for patients with hypertrophic cardiomyopathy. Thank you.
All right, let's hear it for Giska from the Center for Engineering Mechanobiology. Thank you, Giska. That was great. And we'll check back in with a couple of our judges now. So Melissa, what did you think about that slam?
I really love the different mediums that were used to communicate the message, and you can really tell that your passion shines through your words. I mean, the emphasis on some words, the pauses, the different props, it was really well done. Good job.
Thank you, Melissa.
Great. And Jorge, any feedback to add?
Yeah, I thought that was really fantastic. I was delighted all the way through. I had a big smile all the way through. I really liked your energy and your sort of positivity, and I love the way you engage the audience, like asking questions, and then you actually like pause for people to think about it. That's really important. I think a lot of times when we present, we sort of are in a rush, and so that pacing is really good. And like Melissa said, I really loved how creative you were with the space that you had. And I was like, oh my goodness, she's here, she's there, and she's using her whole body to illustrate things. And that was really delightful, as I said.
Thank you, Jorge. Really appreciate it.
Wonderful. Thank you, judges. And really nice job, Giska. Thank you. Now it's time to hear from slammer number 4. Heading into the second half of our program this evening, Sabrina Alcasis is our next finalist, and she is joining us from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution, where she is a student connected to the Center for Dark Energy Biosphere Investigations, which is exploring microbial life in the deep biosphere, the rocks and sediment that make up over 70% of the Earth's surface. Welcome, Sabrina.
space geysers. And these space geysers gave us a window into detecting what its alien ocean contains. Turns out that it has all the right stuff to support this sort of life form: hydrogen, its air, and carbon dioxide, its food. I hope that by studying these amazing microbes here on Earth, we can begin learning about what sort of life could exist beyond. Thank you.
Let's hear it for Sabrina from the Center for Dark Energy Biosphere Investigations. Thank you, Sabrina. Great job. All right, now let's see what a couple of our judges thought about that. So Raven, what's on your mind after hearing that slam?
Well, you did a wonderful job presenting the information. I especially loved the graphics and your sense of confidence with your information. I definitely felt like you knew exactly what you were talking about, and it was really easy to follow along with what you were saying. Well done.
Thank you.
Great. Emily, how about you?
I mean, obviously, this spoke to my little spacey soul. I am very excited about this work, and I think you told it so well. And I loved your comparison of things that are very small to things that are very big. I think you made the correct assumption in that we all probably thought that that was the night sky. And I loved that you flipped it on us. And I think this is all very, very exciting and cool. Well done.
Great. Thank you, Emily. Thank you, Raven. And great job, Sabrina. All right, now we're going to take a look at Slammer number 5. Next up, we have Ben Pingo, a postdoctoral fellow at Harvard University, representing the Center for Integrated Quantum Materials, which is working to create atomic-scale building blocks that could pave the way for powerful quantum computers and communication systems. Take it away, Ben.
Computers are everywhere these days, from our smartphones, laptops, all the way to supercomputers. And they help us so much processing information, solving complex problems. So there's a big incentive to improve them and make them even more efficient, right? Well, what if I told you that maybe one way of making them much more powerful was to use the weird rules of quantum mechanics? The weird laws that rule over the world of tiny objects, such as atoms and electrons. But now, what if I told you that this computer of the future using quantum mechanics could actually be made of diamond? Let me tell you. But first, I need to shrink myself down to the size of a much smaller than a grain of sand, all the way down to the size of a single atom, those tiny particles that constitute all matter. And now I'm ready. Let me jump into the diamond. And now I'm surrounded by all the carbon atoms that constitute the diamond. But I'm not a carbon atom. I'm another type of atom. I'm special. I'm a tiny defect. And I have this very peculiarity. I have a tiny magnet with me. It can be pointing upwards or it can be pointing downwards. And that could be used to store information, like in today's computers, in successions of zeros and ones. So upwards could be 0 and downwards could be 1. But that's a special magnet. That's a quantum magnet. And in quantum mechanics, it says that something can be in several states at the same time. That's the spooky thing about quantum mechanics. So my tiny magnet could be pointing upwards like a 0, pointing downwards like a 1, or both at the same time. Aha. But what does it have to do with computing then? Well, if you have a normal computer program, like in today's computers, you want to know what it does. And so what you do is you have an input and you feed it into the program and you look at the answer. Then you feed another input into the program and you look at the answer. So you see, it took me 2 turns to know what the answers are. But with a quantum computer, you can feed both at the same time and you get the answer. So just one run. So if you have 50 inputs, say, you can all process them at the same time. So you speed up things so much that a very, very complex problem that would take years to solve on the best supercomputers today could be just solved on a quantum computer in a fraction of a second. And that has huge implications, especially to solve very complex problems, such as predicting reliably the weather forecast, understanding complex biological molecules and how they interact with each other, or even optimizing air traffic in real time, which is very complicated. So let's get back to our diamond. Obviously, I can't do all of this all by myself with my tiny magnet. I need other ones like me to process all that information. And look, there's another one over there. But how can we communicate? Well, because diamond is transparent, you can actually use light. And this way, you transfer information between those tiny magnets across one defect to the other, and all together, we can compute information using quantum mechanics and speed all this up. So to conclude, the diamond is just like people. It's their defects that makes them interesting. Thank you.
Let's hear it for Ben from the Center for Integrated Quantum Materials. Thank you, Ben. Well done. And our judges are back too, so let's see what they're thinking. Melissa, what are you thinking about that slam?
I loved the enthusiasm, the pacing, how you made it really relevant to us. And you brought us inside something so small and made it just a concept that was easy to understand. So thank you so much. That was lovely to watch.
Thank you very much.
Great. Thanks, Melissa. And Jorge, any thoughts on that?
Yeah, that was great. Ben, I would give you a 5 diamond rating for sure for that presentation. I like Melissa said, I loved your enthusiasm. You were very animated and very engaging. That was really great. You brought a lot of energy. And again, I saw your presentation last time, but I was still delighted this time with how you really thought creatively about putting yourself into the presentation and interact with the graphics. I thought that was really fun. I think in science communication, a lot of it is about exploring the medium that you're in and thinking creatively about how you can use it. So kudos for that.
Thank you very much.
Great. Thank you, judges. And great job, Ben. All right, now let's take a look at our final slam of the evening. And last but not at all least is Megan Chong, a student at the University of California, San Francisco, and she's affiliated with the Center for Cellular Construction, which aims to develop a new approach to understanding and designing cells that can one day serve as living factories and building blocks for better and more sustainable products and devices. Over to you, Megan.
We are all made of about 30 trillion cells each. And every day, 2 trillion of them do something called cell division. The purpose of cell division is to make more cells as your body grows, and later to replace cells you've lost. So that's something like 700 trillion divisions happening in your body every year. And today, I want to talk about the challenges cells face every time they divide. Most of you have heard of DNA. DNA gets folded up into chromosomes, and it gives your cells instructions on how to make you. And it's really important that each new cell you make has a full set of instructions. One chromosome looks something like this. And just before a cell divides, it makes copies of all its chromosomes and sticks the copies together like so. And the key challenge during cell division is to make sure these copied chromosomes are split evenly into 2 new cells. So let's zoom in and look at a tiny living cell. This cell, outlined in purple, has its chromosomes at the center, and a football-shaped structure called the spindle is in green. And as I play this movie, you'll see that the spindle is a structure the cell builds that attaches to chromosomes and pulls them apart as the cell divides. And building that structure well is really important for your health. Failing to separate your chromosome pairs or letting cell division get out of control can lead to diseases like cancer. So how do cells actually solve this technical problem of engineering a spindle that can divide your chromosomes so well every time? You might think that, like an engineer, they follow some sort of blueprint or detailed instructions, like you'd find in a Lego set that say, attach chromosome 1 to both sides of the spindle, now chromosome 2. Instead, the cell takes a much more winding path through division, and that helps make chromosome separation more accurate. You can think of it like solving a maze. If I plopped you down in this maze with instructions that told you which way to go at each fork, you'd cut a clean path through. But then, if you tried to solve a new maze with those old instructions, you'd get stuck pretty quickly. So what if instead I could give you a trick or a rule to follow that would get you out of any maze? You might have heard of the right hand rule. You can find your way through a maze by keeping your right hand on the wall beside you as you wind your way through. Now, your path is a lot more roundabout, but you'll definitely make it out. So back to our dividing cell. As cells build the spindle, they make some wrong turns. Sometimes a chromosome pair doesn't touch the spindle at all. In this case, the rule is that chromosomes send a loud chemical signal saying, "We're not attached," and the cell waits for the spindle to correct its mistake. Other times, both copies of the same chromosome pair attach to just one side of the spindle. I'm trying to figure out the rules that help the spindle correct this mistake. By learning the rules cells use to build the spindle and other structures, we can improve our understanding of diseases like cancer and design treatments. But we also hope that with these rules, we can begin engineering better versions that avoid mistakes altogether. Thank you.
Just want to give you all a heads up.
All right, now let's hear it for Megan. Great job, Megan. Thank you so much. It was Megan from the Center for Cellular Construction. And now let's check back in with our judges. So Emily, any thoughts to share on that?
I love that. That's my first time seeing that one, but I loved the way that you told that. It was clear that you were very confident in your delivery, and that is such an important part of all of this, because I need you to be confident in what you're telling me so that I can be confident in believing you. And it was so good. It was clear that you practiced it and it was well delivered. Good job.
Great, thanks, Emily. So Raven, what's on your mind after that?
Well, as a molecular biologist, I have to say I really enjoyed it, and you did a great job in finding analogies in real life. I loved your maze example. I had no idea that you could run your hand on the wall and find an easy way out. I want to try that now. And every time I see a maze, I'm going to think about this presentation, which is amazing. I did learn something new, and I thought it was super engaging. You did a great job. And as Emily said, very confident. Well done. Well done.
Thanks so much.
Great. Thank you, judges, and thank you, Megan. Great job. Now, it turns out we had a little bit of an audio glitch during Sabrina's presentation, so we're going to bring her back on to do it again. So I am going to turn it over to Sabrina. Heading into the second half of our program this evening, Sabrina Alcasis is our next finalist, and she is joining us from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution, where she is a student connected to the Center for Dark Energy Biosphere Investigations, which is exploring microbial life in the deep biosphere, the rocks and sediment that make up over 70% of the Earth's surface. Welcome, Sabrina.
Imagine you are deep in the heart of Russia, trekking through these vast mountains, only to find this. A lake so toxic to life it seems almost extraterrestrial. This isn't sand that you see here along the shore. It's mineral deposits. Nothing should live here in these waters containing a huge amount of chemicals, like baking soda, which make it seemingly uninhabitable for life, right? Wrong. There are tiny, tiny microbes called archaea, which populate this lake. Now, they don't breathe air like you and me. They breathe hydrogen. They don't eat hamburgers and mac and cheese like you and me. They eat carbon dioxide. They exist outside the conditions considered normal for life, almost like aliens, right here on Earth. In my lab, I began simulating the conditions of this lake in this little vial. That's right, folks. In this vial, there are billions of microbes. Billions. But why bother? It's cool and all that I'm growing these, but why go through the trouble of growing them in the first place? I want to bring your attention to this picture here. I will give you a few seconds to come up with a guess of what you think it is. Okay, do you have your guess? Well, if I had to guess your guess, I would guess that you think it's the night sky or outer space, or in other words, really, really big. It is in fact not outer space. It is microbes under the microscope. Each one of these tiny blue dots is a microbe. However, I am growing these microbes to study outer space, so you weren't wrong at all if that was your guess. I am using these microbes to study what signs of life we should look for if we send a probe to explore the deep and ice-covered oceans of several moons in our solar system, which are shown here in comparison to Earth. The oceans on these moons are out of the range for sunlight to provide energy for life, and thus any organisms that live there would have to survive on chemical energy, just like the archaea do in the toxic lake here on Earth. I'm particularly interested in one of Saturn's moons, called Enceladus. Now, Enceladus has one of these cold and icy oceans, but it also has space geysers that gave us a window into detecting what its alien ocean contains. Turns out, that has all the right stuff to support this sort of life form. Hydrogen, its air, and carbon dioxide, its food. I hope that by studying these amazing microbes here on Earth, we can begin learning what sort of life could exist beyond. Thank you.
All right, thank you again, Sabrina. That was wonderful. And now all of Sabrina's lovely images and the judges' comments all make sense. Now that you've had a chance to hear her wonderful slam, thank you for that. And so now we've completed our slams. And so, judges, you have been through the semifinals with these incredible scientists. You've had a chance to share this journey with them. Do you have any thoughts to share with the slammers and our audience before you go into your deliberation room to finally select a winner? So, Melissa, let's start with you.
I think probably the thought that all of us are kind of feeling is this is going to be a tough decision. These are all such amazing presenters. They've really made us think, be engaged, learn something new. So yeah, it's definitely going to be a tough decision.
Absolutely. It's going to be such a tough decision. So, Raven, what are you thinking?
There's so much talent here. It's incredible. Everyone did an extraordinary job. And as we've said in the past rounds, we have to pick someone, but we are so excited to see what everyone is bringing to the table and how you're moving science communication in a forward direction, in a more innovative direction. Keep up the great work. And no matter what happens, y'all are wonderful. Congratulations.
Thank you, Raven. And Jorge, how about you? Any words to share?
Yeah, I am definitely slammed by science all around. I thought everyone was great. I noticed everyone dressed up a little more for the finals, so kudos for that. But yeah, it was just great to see how a lot of you evolved, for instance, the last time we talked, and just it seemed like everyone was just more confident and more determined this time. So.
Definitely. Thank you. And Emily, any final words to share before you go off to make your big decision?
Yeah, I mean, I think that one of the takeaways I'm finding here is that there is such a diversity in communication styles in all of these presentations. And I love that because as science communicators, we're trying to reach a diverse audience, and one communication style just isn't going to cut it. And so I think a good thing to keep in mind is that whoever wins tonight, that's not to say that that's the communication style that's good. That's just, whoever wins tonight. I think that one thing that I love is that all of you did a really, really good job and staying true to yourself, and knowing that you are going to reach an audience that I wouldn't be able to reach, or Jorge wouldn't be able to reach, or Melissa or Raven, any of us, And so I think staying true to yourself and just practicing that communication style is going to be really, really worthwhile.
Great. Thank you so much. And now it is time. We're going to send you off to go make your decision. So we'll see you soon. Thank you so much, judges. I do not envy the decision that you need to make. Everyone is a winner tonight. They did such a fantastic job. Now, if you're wondering how the judges are evaluating the slammers tonight, well, we asked them to consider 4 judging criteria as they select their choice for the grand prize winner. And they are scientific content, creativity, clarity, and one more very important element, their connection to the audience, you. Okay, now slammers, you all did such an amazing job tonight, and you've come so far. I do not know how our judges or our audience are possibly going to make a decision. To us, you are definitely all winners for being here and for sharing your amazing work with us. Now, I would love to hear your thoughts quickly before the audience voting begins. So how are you all feeling about the slams tonight? Any thoughts? Jolene.
It feels like we've come to the end of this journey that we've all traveled together. And I was so excited to see everybody else and to see how, basically we've all evolved together. And so it was really lovely. Yeah. I'm so glad I got to do this with all of you. And I'm really happy for this experience.
Thank you, Jolene. We're very glad to have you. Any other thoughts about the competition before we launch the audience voting?
Yeah, I also learned a lot from the other slammers. You guys, you did a great job. And I watched your semifinals as well. And I see how all of us working, try to improve our slams. And this is really great experience for me. Thank you.
Wonderful. Thank you so much, Giska. All right. Any final comments? Okay. Well, it is time to start the audience voting. So here we go. Now it's your turn. To vote for your favorite science slammer, go to pollev.com/Museum of Science and select their name from the options there. Or if you'd rather vote by text message, text Museum of Science to 22333 and then send us the number on the screen next to your favorite slammer. Those numbers are 1 for Jolene, 2 for Christina, 3 for Giska, 4 for Sabrina, 5 for Ben, and 6 for Megan. The clock is counting down from 5 minutes, so make sure to get those votes in. And while the voting is going on, we'll be showing some of the comments that are coming in from our audience to cheer on our slammers. Two minutes to go on the clock. Keep those votes coming. You can vote online by going to the web address on the screen, pollev.com/Museum of Science, or you can text Museum of Science to 22333 and then send us the number next to your favorite slammer. Those numbers, one more time, are 1 for Jolene, 2 for Christina, 3 for Giska, 4 for Sabrina, 5 for Ben, and 6 for Megan. One more minute to vote. And we are done. All right, so the audience voting has now ended. All of the prior weeks have led us to this moment. We are about to announce the first of our 2 winners tonight. Are you all ready? All right, our audience choice winner is Giska! Congratulations, Giska! You will receive a $1,000 prize from the Museum of Science. Congratulations, fantastic job. So I'm going to give Giska a chance to come up here and join me. In a second, we will bring Giska up on screen, hopefully sometime soon, to ask her how she's feeling. There she is. Giska, I don't think you can see me anymore, but I'm here. Hopefully you can hear me. So how are you feeling? Congratulations.
Thank you. And I would like to thank all of the audience and the voters, because without your support, I won't be here.
Wonderful. Well, we are so happy for you. Congratulations. So now that you've been through the Reach Out challenge, what do you think you'll continue on with science communication?
Yeah, I really remember that the first time I joined the Reach Out Science Communication Workshop was last year, like December 2020, when I was like down because it was in the middle of the pandemic, and it was also my second year of my PhD, also my second year in the US, where the English is my language barrier. So I can't believe, but the workshops really helpful for me, especially how you, like Megan, Meg, Carrie, and also Caroline, like were really positive, and I really absorbed that positive energy, and I just getting through the process, just enjoying it, how you coach us, and yeah, I really enjoyed it, and I feel like I found my passion.
Oh, that's wonderful. Thank you so much, Giska. That is so wonderful to hear. You did such an amazing job tonight, and we are going to hear more from you in the future. Thank you so much. And now our judges are coming back. Welcome back, judges. How are you all feeling? So, Raven, you are our spokesperson for the judges tonight. Do you have any comments before you make your big announcement?
I would like to say that all of the judges agree that each and every one of you did a spectacular job. And for the presentations that we have seen, we can tell that you all have been practicing and working hard on perfecting your presentations and your craft. We were talking about behind the scenes, we want each of you to start developing your own platforms and making sure that you continue on your journey as science communicators. You're doing excellent work. And our decision tonight does not devalue any of the hard work that you've already done and that you will do in the future. So you can all be proud of yourselves. And with that being said, I think we're ready.
We are ready. Raven, who have you all chosen to receive our second prize?
Christina.
Christina, congratulations. Oh, there she is. She's up here already. Amazing. Congratulations. So, Christina, how does it feel to be our other Reach Out Science Slam winner?
Oh, it's so very exciting. I had so much fun listening to everyone else's too. I'm sad we couldn't all just keep going and keep presenting more.
That's wonderful. So, what inspired you to enter the Reach Out Slams, Christina? What got you into this?
Yeah, I mean, I've had a lot of practice explaining science to, my little sister who teaches math, or my mom who does math, or my brother who does like video filming stuff. So I figured, why not use some of that experience to explain science to more people?
Wonderful. Well, we are very glad that you did. You did an amazing job. Congratulations. Congratulations to both of our winners, Giska and Christina. You will each receive a $1,000 prize from the Museum of Science. Nice job. But congratulations to all of our finalists who presented tonight. You did an absolutely amazing job, and you will all receive $250 stipends to thank you for working so hard to share your science with all of us. I'm sure we will hear lots more from all of you in the future as you progress in your careers as scientists and as you continue to reach out to public audiences. And thank you again to our absolutely incredible rock star panel of judges for sharing their experience and their expertise. They have been so supportive of our slammers throughout these events. We cannot thank them enough. They've been just absolutely amazing. All of their comments, their advice, their feedback. It's been really, really great to have all of you here, thank you so much. It's been an honor. I want to give all of you a chance to wave goodbye to our slammers and our audience. Thank you so much, judges. All right, and that brings us to the end of the Reach Out Challenge. It's been an absolutely incredible journey. Thanks so much to all of you for joining us for the slams, and thank you to our sponsors, the National Science Foundation and the Center for Integrated Quantum Materials. And we would love to hear your thoughts on this event. If you would like to tell us what you think, please fill out a brief survey using the QR code up on the screen, or you can find the link at mos.org/reachout. We will randomly select 5 online audience members who complete the survey to receive a $50 Amazon gift card. All right, congratulations again to all of our wonderful slammers and judges and our sponsors. Thank you again to all of you for joining us, and have a wonderful evening. Bye.